Acid-base balance. We will be looking into the normal acid-base homeostasis of the body, the role of first line and second line and third line buffer mechanisms for the regulation of acid-base balance. We will be looking into the acid-base disorders and A9 gap in the next session. So starting from our first topic of discussion, we will first look into what are acids and bases and related concepts. So a hydrogen ion is a single free proton released from a hydrogen atom. And acids are the molecules that contain this hydrogen atom and can release hydrogen ions in a solution. Examples, HCl, carbonic acid, whereas base is a molecule that can accept a hydrogen ion. Examples like bicarbonate, phosphate ions, and even proteins like haemoglobin can also accept hydrogen ions acting as bases. Bases and alkali are used interchangeably. However, alkalis are formed when the alkaline metals combine with the hydroxyl ion, for example, sodium hydroxide. If there is excess of hydrogen ions in the body, we call it acidosis. If there is excessive removal of hydrogen ions from the body fluids, we call it alkalosis. Whereas pH is a negative logarithm of hydrogen ion concentration. Normal hydrogen ion concentration in the body, 40 nanoequivalents per liter, range may be 35 to 45. pH average 7.4, range 7.35 to 7.45. Less than this is acidosis, more than 7.45 is alkalosis. This is comparing the values in the extracellular fluid versus other fluid compartments. Interstitial fluid and extracellular fluid approximately having the same pH. However, this is little less in the venous blood as compared to the arterial blood because of the presence of more carbon dioxide. Intracellular fluid pH is having a higher range from 6 to 7.4, urine 4.5 to 8, and gastric pH is much more acidic, as we know. Acid-base regulation is done by three lines of defense, the first one being chemical acid-base buffers, second respiratory center, and the third kidneys. If we compare these three, the chemical acid-base buffers immediately combine with the acid or base to prevent change in the pH. They cannot, however, remove acid or base. Respiratory center can remove excessive carbon dioxide or retain it to maintain the acid-base balance, whereas kidneys can exactly remove the acid or alkali to adjust the extracellular pH. The chemical buffers respond within seconds, respiratory centers require minutes, whereas kidneys requires hours to days to complete their action. Chemical buffers act as the first line of defense, but they just minimize the change in the pH, cannot eliminate the cause. Respiratory system act as the second line of defense, much more effective than the chemical buffer system, but the most powerful are still the kidneys, which can permanently eliminate the excessive acid or the base. To discuss the chemical buffers for the acid-base balance, the first line of defense, let's just first see what buffers are. Buffer is a chemical substance that can reversibly combine with hydrogen or hydroxyl ions to maintain the pH stability. For example, the most important buffers in the blood include bicarbonate, protein buffers, and the hemoglobin buffers. In the interstitial fluid, most important, again, bicarbonate buffers, whereas intracellular important buffers include protein and the phosphate buffers. This is showing the titration curve of a buffer system. On x-axis, you can see the pH. On y-axis, you can see the relative concentration of the acidic and basic components of a buffer. Here is the example of the bicarbonate buffer system. Important thing to understand over here is the concept of the pK. What pKa is the pH of the system at which the both acidic and basic components constitute 50% of the total concentration of the system. As you can see, 50% and 50% of both constituents, and the pK at this point will be known as pK. The buffer system is most effective in the central part of the curve. As you can see, there is small change in the pH by regulating the change in the relative concentration of the buffer systems. However, at the extremes, the buffering power of a buffer system becomes less and the pH change is more with a small change in the relative concentration. So, two takeaways from over here: pK is the pH at which relative concentration of both substances is 50%. Number two, the buffers are most effective when pH of the system is close to the pK. This is the example of bicarbonate buffer system and its dissociation constant. For any acid, the concentration of the acid relative to its dissociative ions and Henderson-Hasselbalch equation can be used to estimate this dissociation constant. Buffer power of any buffer system is determined by relative concentration of the buffer components. As we just discussed, the buffer power is going to be more when pH of the system is close to the pK of the buffer system. Number two, other important thing is absolute concentration of the buffer components. For example, there are two buffer systems, both having pK, let's see, one is 7.5, other having pK 7.6, buffer system A, buffer system B. And the pH of the solution is 7.4. Okay, let's make it 7.8. Now, the pH of buffer system A is more close to the pH of the system, so it is going to be more effective than the buffer system B. This is the role of relative concentration. But we have to consider the absolute concentration as well. The pH of buffer system A, pK, is close to the pH, but the concentration of buffer system, if we say, is 1 and concentration of buffer system B is 10 times more. Now this is 10 times more, so this is going to be the more effective buffer. So we have to consider the both of the things. For a buffer to be effective, its absolute concentration would be in a good amount, plus its relative concentration of components should be close to the 50%. The buffer efficacy determined by the relative concentration as well as the absolute concentration. If we apply this rule to the bicarbonate buffer system, the pK of the bicarbonate buffer system is 6.1, whereas the pH of the plasma or extracellular fluid is 7.4, which is quite away. So not in favor of it. And if we look at the absolute concentration of the buffer components of the bicarbonate system, that is also low. So the two conditions that we had determined for efficacy of a buffer, both are not qualified by the bicarbonate buffer system. Still, this is the very important and in fact the most important buffer of the plasma and the interstitial fluid. And why is that so? The reason behind is that bicarbonate buffer system has two components, the bicarbonate and carbon dioxide, regulated by the kidneys and by the lungs. So we had three lines of defense: buffers, kidneys and lungs, and the components of the bicarbonate buffer systems are directly regulated by the other two buffer system defense systems of the acid-base balance, making it still the most important powerful extracellular buffer. So to summarize, a buffer is going to be most effective in this range close to the pK, but not only its relative concentration of components, the absolute concentration also matters. A buffer system may be more effective in one fluid system and less effective in the other. For example, phosphate buffer system is more effective intracellularly and bicarbonate buffer system extracellularly. So let's compare these three important buffer systems. Bicarbonate buffer system, more important in ACF, components, weak acidic component, carbonic acid, and basic component, sodium bicarbonate. pK is 6.1, responds to acids and base by combining with them and minimizing the changes in the pH. Phosphate buffer system, most important in renal tubular fluid and intracellular fluid. phosphoric acid and hydrogen phosphate. Its pK is 6.8, which is close to urinary pH, responds to acids and bases by combining with these and minimizing the change in the pH. Protein buffer systems important in intracellular fluid. For example, hemoglobin buffer system, the ammoniamines, pK is close to the ICF pH, so more effective over there, respond to acids by combining with them. Isohydric principle states that all buffers in a common solution are in equilibrium with the same hydrogen concentration. It's not like that bicarbonate buffer system is more effective, so first it will respond and then the protein buffer system and then the hemoglobin buffer system. Rather, all the buffer systems present in the solution will start working equally to respond to the change in the pH and come in equilibrium to it. That was all about the first line of defense for the acid-base regulation. Now the second line of defense, which is respiratory regulation. How do the respiratory system respond to it? We can describe by using these graphs. As we can see, the change in the pH on the x-axis and alveolar ventilation on the y-axis. Normal operating point in the body is between approximately over here 7.4 when the alveolar ventilation rate is 1. As the pH keeps on decreasing, more hydrogen ions keep on accumulating, the rate of alveolar ventilation keeps on increasing to excrete more carbon dioxide. So the respiratory system responds to acidosis by hyperventilating and alkalosis by hypoventilating so that carbon dioxide can be retained. And this is showing the same concept. On the other hand, if for any reason rate of alveolar ventilation will increase, the pH will increase in the body. If rate of alveolar ventilation decreases, pH decreases. The rate of alveolar ventilation can be increased as low as 0, maybe stopped, or as much as 15 times normal. So by looking at this fact, can you guess the correction for acidosis is better or correction for alkalosis is better? For acidosis, the respiratory system has to hyperventilate. It can increase its rate of ventilation from 0 to 15 times. Whereas for alkalosis, it has to decrease from 1 to 15. For alkalosis, it has to hypoventilate and it has a very less capacity to hypoventilate as compared to the very high capacity for hyperventilation. So the correction for acidosis is going to be more effective as compared to correction for alkalosis. Respiratory regulation for acid-base balance via lungs by increasing or decreasing the rate of alveolar ventilation. Alveolar ventilation means the respiratory rate multiplied by the tidal volume minus dead space volume. Effect of low alveolar ventilation will be retention of carbon dioxide and increased hydrogen ions. The buffering power of respiratory system is about 1 to 2 times more as compared to the chemical buffers, although the chemical buffers are much faster. Its efficiency is 50 to 75%, that is a feedback gain of 1 to 3. Response in 3 to 12 minutes, and if respiratory system is impaired, leading to less effective exchange of gases, it may itself lead to respiratory acidosis. Respiratory compensation for metabolic acidosis is by hyperventilation, which excretes the excessive carbon dioxide, and for metabolic alkalosis is hypoventilation to retain the carbon dioxide. The respiratory compensation for alkalosis is not much effective as compared to acidosis. The reason, one, it can increase its respiratory rate from 1 to 15 times but decrease only up to 1 times to 0. Number 2, when the respiratory system is trying to correct for alkalosis, not only carbon dioxide is being retained while oxygen deficiency is also occurring. If significant hypoxia develops, this will overcome the drive and respiratory activity will restart to account for the hypoxia instead of responding to the acid-base imbalance. That's all about the second line of defense for the acid-base balance. We will continue with the third line of defense in the next session.